Today I'm going to talk to you about some of the work that we've been doing in our little research group by the state. We're taking two waste materials, lactic acid, which comes from, as you'll see, yogurt production, and green crab shells, which again is a invasive species that's generally a waste product on its own. It's basically describing a paper that we published earlier, late in last year. So I'm a chemist. I'll speak slowly. We're focusing on some of the real big challenges that we have here in Canada, including efficiency in fuel use, manufacturing advanced materials, harnessing bio-waste, looking at wood chemistry, and focusing on some real important environmental technologies such as carbon capture, mercury cleanup, CFL recycling, and bitumen processing and cleanup. That's where we make, that's our bread and butter for our group, is working on these environmental technologies. And, you know, grew up on a farm. I know exactly what it's like to grow up in a very rural community, so I'm glad to see some of the work that Tony does, because it's very challenging to live in some of those areas and make a good living. And of course, I really appreciate the fact that one man's waste is another man's gold. And here, this is a polymer isolated from crab shells. It's a chitosan. It's the stuff that people take for joint pain and things like that. And it has various functionalities on it. It's a polymer, second most readily available biopolymer. And it has all kinds of useful applications. But the problem is that in all industrial processes that isolate this valuable material, we use hydrochloric acid to get it out. Chitin can make up the crab shell or lobster shell up to about 15 to 20%. And getting that material out depends on having a good and easy and cheap source of HCl, which, for those of you who know the chemistry, is not a very green process to get. And as we transfer into a lower carbon environment and economy, access to HCl is going to get more and more and more challenging. You probably have seen this little guy. It's the European green crab. They're appearing all around Nova Scotia. I actually don't really fully appreciate this problem. I just know that this is a raw source of chitosan, and that's the way I looked at it when we were given this challenge. And it's a bycatch, and it is valueless as a source of food, and you're not even allowed to throw it over the side of the boat when you catch it. You have to keep it and then dispose of it. So it's really a true waste product, and it's ready to be used here in Nova Scotia. The problem is, of course, you have to throw it in with some HCl. So what we said, well, in order to demineralize this and replace the hydrochloric acid, we figured, well, let's see if there's another acid that we could use to do the same chemistry. And that's where we looked at lactic acid. Lactic acid is available in multi-million tons in North America. And that's because it's used to make, it's a byproduct of cheese making. And diabolically, the biggest source is from our dependence upon Greek yogurt. Because when you make Greek yogurt, you end up throwing a lot of the whey away, if that makes any sense. And it's a waste. And the waste from these plants is so acidic that you can't dump the whey into the typical municipal waste streams. So down the valley at various farms down there, you can't dump it into the municipal waste. What you have to do is you have to dump it right onto your farm. And that creates all kinds of problems. So if we can find a way to link the green crab up with the waste from the cheese making process, we might be into something pretty interesting. And like I said, it's dirt cheap. It's about $500 a ton, depending on industrial conditions in North America. So it's really cheap. So we take the little bits of crab shell. They actually look like those ears that they grow under people's skin, right? But it's a very fuzzy picture, and I apologize for that. And you take that and you immerse it into lactic acid. You see the bubbling that you would expect. You get carbon dioxide bubbling off. And slowly the crab shell gets thinner and thinner until you're left with a wafer or like a leather sheet of almost pure chitin and chitosan. works really well. We do chemistry, so I'll show some chemistry. This basically shows you the best concentration to do the reaction in its fastest, around 1 to 5 dilution ratio, which corresponds to 1.9 molar lactic acid. It goes all the way to completion, so there's no waste except CO2 and water. Various crab shells after the process showing behavior. And the chitin that's produced at the end of the day is still very good, high quality chitosan, ready to be deproteinized with strong base. That's too much. We followed the extent of demineralization, and this is just showing as a function of temperature, and it works reasonably well at room temperature, which is a good thing, slightly better at elevated temperature, but the cost saving at working at room temperature, 20 degrees Celsius, is much better than working at 60. So it looks like this is a viable process. The cool thing is, the cool thing is, got this beautiful crystal structure, and why I'm going to say that is that's so beautiful is that this simple complex is actually the crunchy bit that corresponds to the calcium in cheese. This was the first report of this structure and nailed that long-standing problem down. And I'd just like to thank the people that fund our research, some of the big industries in Canada, also Environment Canada, NCANA, NSERC, and various other funding agencies and companies as well. Thank you.